everybody. My name is Frank Duong, and I am a professor in the Department of Biochemistry at the University of British Columbia, and I'm located in the Life Science Institute. So there, my lab is studying membrane protein. First of all, membrane protein are directly exposed to the cell surface, so therefore they are prime target for drug discovery and antibody development. However, membrane protein are low abundance, difficult to express, difficult to purify, and difficult to work with. The reason being that membrane protein are perfectly water insoluble. So therefore, biochemists need to develop solutions to keep them in a water-soluble state. So quite often, we use detergent. Detergents are great to extract membrane protein. However, they have a tendency to over-delipidate membrane protein, and in consequence, detergent easily destabilize protein structure. If not, they definitely destabilize membrane protein complexes into single subunits. The major drawback of detergent is that they always need to be present for whatever downstream application, which creates difficulties. So for that reason, a number of laboratories, including ours, have been developing what we call membrane mimetics that allow to bypass this negative effect of detergents. They are amphipole, small polymer, nanodisk, and picodisk. Nanodisk is by far the one that has been the most developed, including by our lab, to study membrane protein. Yet there's a number of caveats. And to circumvent them, we more recently developed a small amphipatic peptide that has the ability to displace detergent and bind at the surface of membrane protein in order to keep them in a water-soluble state. And we call that a peptidisc particle using a peptidisc peptide. Okay, so this is a cartoon, but we got some structural evidence using cryo-electron microscopy that indeed our peptidase peptide here in pink is binding well at the surface of membrane protein. That was in the case of the ABC transporter MSBA, and this is in the case of the mechanosensitive channel MSCS. And what is striking to observe here is that the peptide is not adopting a fixed conformation, especially in the case of MSBA. You can see some peptide being tilted, organized on different way at the surface of the target. And that is important because it's really the base that explains the adaptability of the peptide for different membrane protein. And using this very interesting property, we have been developing more recently what we call a peptidase library, which is the capture of the whole membrane proteome in a water-soluble state using this peptidase scaffold. So here you have membrane protein, many different types of membrane protein with cytosolic components, follows solubilization by a detergent and replacement by the peptidase scaffold. It is possible to transform this membrane protein in a water-soluble state. And this soup of protein can be directly analyzed in the by mass spectrometry. And indeed, we can see that in the top 100 protein identified, we have about 20-25% of them being membrane protein, which is expected, a normal proportion for membrane protein in a total cell extract. A trick that we apply here is to use a peptidase scaffold that carries a tag so that we can specifically enrich our library for membrane proteins. And indeed, if you follow the same mass spec analysis, in that case, the top 100 protein ID, more than 80% will be membrane protein. So qualitatively, we have a significant enrichment, but also quantitatively, the intensity of the peptide we detect on the mass spec is much higher, indicating an effective enrichment. So that was initially developed using E. coli membrane proteome, but we have also been using mammalian cells culture as well as tissue such as brain, heart, and kidney. So we capture membrane protein from the different compartments, cell membrane, ER membrane, mitochondria, and the various organelles. So there is not specific bias here. Importantly, if you look at the nature of the membrane protein being captured, we capture membrane protein having one transmembrane segment from 10 up to 22. And what's important to note is that this particular distribution is actually has been observed in transcriptomic analysis as well as other destructive proteomic analysis. I say destructive because this one use SDS in order to identify the membrane proteome, whereas in our case, we are recovering a protein in a functional state, water-soluble state. So what type of protein do we recover? If you group them by family, we have a lot of membrane enzyme, a channel, receptor, SLC transporter, P-type transporter, ABC transporter, CD antigen, and so on. This is in the heart. In the brain, we capture more channels as well as expected GPCRs, whereas in the kidney, we capture more SLC transporter in that case, which is in relation to the function of the kidney. So overall, I think we have a faithful capture of the cell membrane proteome in water-soluble particle. So what can we do with this peptidase library? That's why it become important, I believe, is that we already have membrane protein in water-soluble state. So we can look for a change. Since this protein can be easily detected in a mass spec, we can detect low abundance, but also change in the proteome response as a function of drug or even identify biomarkers. In the context of protein purification, since we have water-soluble particle, the classical fractionation workflow, IEX, GF, affinity purification can be applied. We can use the library for screening phage and yeast display library, as well as identifying off-target antibody, which is important in the context of preclinical assessment. And finally, most importantly, we can really start to study protein-protein interaction, protein-ligand interaction in the total absence of detergent. And for instance, we can look for novel receptor-ligand interaction, as well as screening library of small molecules. Okay, so here, following this short presentation, I want to thanks previous and current member of our laboratory, our federal sponsor for this work, as well as collaborators, and also thank you for your attention.